Hello, welcome to this session on data science and neuroinformatics. My name is Ariel Rokem, and I'm a research assistant professor and a data scientist at the Department of Psychology in the eScience Institute at the University of Washington, where I lead a research group in neuroinformatics. In today's session, we will hear several talks that combine neuroinformatics and data science. And in this introduction, I'd like to present to you a few ideas about why we need data science, what data science is, give you a little bit of a historical perspective about how we got here to the point where we're talking about data science in the context of neuroinformatics, and then focus in on what I think are important and salient features of data science, what I call data science in practice. I'll finish with this idea of intellectual infrastructure, the idea that we need to invest in people and think about people rather than machines or data per se when we think about data science. And finally, I'll introduce the speakers for this session. So how did we get here? Well, one way to think about this from this paper from Stevenson and Kerding that has been updated recently is to think about the deluge of data that we're seeing in neuroscience. So this is a chart that shows you the publication date on the x-axis of different papers and the number of simultaneously recorded neurons in these papers. And you can see that this has a Moore's law kind of growth with a doubling time of about between 6 and 7 years. And even in recent years, the growth has been even higher with several papers recording from tens of thousands of neurons simultaneously. And this generates a lot of data. And we see this everywhere in neuroscience. I work in human neuroscience, and I work with these kinds of data sets like the Human Connectome Project. It's collected 1,200 subjects, and that's completed. And this increasing growth, we see this in our field as well with projects like the ABCD study and the Child Mind Institute's Healthy Brain Network dataset, collecting ongoing collection of up to 10,000 individuals. Finally, the UK Biobank, really the biggest one that I know of, with data collection ongoing for eventually up to 100,000 subjects, where we'll have really high quality MRI data for all of these individuals. And so this is a massive data, and there's a lot that we can do with this data. And data science might be maybe thought of as all the things that need to be in place for us to be able to analyze and understand all of this massive data that's coming down the pipeline. And that includes things like the computational infrastructure that we need, the statistical analysis techniques that we need, the software, and many other things that I will talk about in just a few minutes. But how did we get here? What's the sort of historical underpinning of this idea? So the idea of data science is not necessarily such a new idea. I think the first time that it was introduced in sort of an academic context, this idea of data science, was actually in statistics. And this is a paper from William Cleveland from 2001, talking about how statistics should expand its field of view to include more technical ideas. And in this paper from 20 years ago, he mentions a few sort of ideas that statistics should incorporate to form an action plan for expanding into technical ideas. And that includes multidisciplinary investigations. So that means collecting data from various fields and looking at that data, what we call subject matter areas here, models and methods for data, all kinds of more sort of core statistics ideas, computing with data, and that's been an important part of data science ever since, and sort of the intersection between computer science and statistics, statistical computing. Puts an emphasis on pedagogy, curriculum planning, and approaches for teaching these ideas. A nice portion is put towards tool evaluation, and that I think is very, for us now in data science, is something that seems very important as the tools for doing data science are evolving so rapidly. And then finally, what you can think of as really the core statistical ideas, the theory, the foundations, general approaches to models and methods and so on. And that's the part that I think of, the statistics part of this plan. So this was 20 years ago, so maybe we've had 20 years of data science. But just a few years ago, actually, David Donahoe pointed out that in a paper titled 50 Years of Data Science, that many of these ideas have been around for a while. He points to this paper by John Tukey in 1962 called The Future of Data Analysis as being really a starting point for thinking about all of these ideas. How do we do data analysis? How do we take statistical ideas from computing and use them in practice. Meanwhile, in Silicon Valley, like many other things, people started reinventing data science as well. So this is a long quote from an interview with DJ Patil, who had gotten his PhD in math and then went into industry to do some work in data. And he mentions that he was at LinkedIn building his data team, and Jeff Hammerbacher, who was the co-founder of Cloudera, was building a data team at Facebook, and they really wanted to collaborate and compare notes, and they weren't sure what to call themselves. And of course, DJ Patil, who was data-minded, tested all the job titles. He put different job titles up on his platform and saw which ones got the most interest from job applicants. Turns out that everybody wanted to be a data scientist. So we're like, okay, that is what we will call ourselves, data scientists. And so this idea, around 2010, this idea started taking hold also in industry, that there is this notion of data science that is something that people who engage with data, maybe at large scale, like the data that these industry players have, that we could call what they do data science as well. And DJ Patil continued on from LinkedIn later to become the United States' first data scientist within the White House. So he took that on a national scale as well. Okay, so what is data science after having said all that? Well, I don't really think of it as a scientific discipline. It might not even really be a profession. Really what I think of it as is an engagement with the practice of data analysis. It's how do we do data analysis? Part of what's interesting about this is, of course, that it crosses disciplinary boundaries. I mentioned statistics, I mentioned computing, and of course, neuroscience, but also crosses sectors. So we're now in the domain of the kinds of things that companies like Google, like Facebook, like Amazon are doing as their core business. So this is really at the core. Data analysis is at the core of these very, very large, some of the largest companies in the world. And that makes for an interesting set of opportunities, but also interesting challenges in engaging with these ideas. In academia, you might think of data science as being at the heart of several different kinds of things that we do in academic research. And here at the University of Washington, this institute that I'm part of, the eScience Institute, which is the hub for data science activities at the University of Washington, we think of this as having three different spokes or fields that we engage in. One is in education and teaching data science. The other is in the forming of a community, outreach, and having working groups of ideas that we engage with. And then finally, the research in data science, using data science to do all kinds of collaborations with people in different fields. So, for us, data science is not exactly computer science. It's not exactly statistics. And for the purpose of our session today, maybe you think of brain data science as being part of neuroinformatics. Maybe neuroinformatics is brain data science. The main idea that I would like to put forth here is this idea of intellectual infrastructure. And I owe this phrase to Ed Lazowska, who is the founding director of the Science Institute here at the University of Washington. And that's the idea that instead of focusing on machines here on the left, this big server farm with the data that is stored in that, really data science is more about people, about their ideas, about the communities that they form around software projects that they maintain, around interactions that people have with each other, and the ability to collaborate with others. So data science is inherently a collaborative practice. And it does involve a variety of fields that we've talked about. It involves statistics and machine learning. It involves software engineering, building stuff. It involves open source software many times. And it involves this idea of communities of practice, the idea that people work together and develop practices together for data analysis. And so with that, I'm opening this session. We'll have three speakers here today that do data science at various levels in interaction with neuroinformatics. Our speakers include Eva Dyer, who's at Georgia Tech, and Eva's work intersects with statistics and machine learning and the analysis of large-scale data sets, a variety of kinds of data sets in neuroscience. And Eva Dyer, I dare say, she calls herself a data scientist. Her lab at Georgia Tech is a neuroscience data science lab. In addition, we'll have Mackenzie Mathis from EPFL. Mackenzie's work involves the development of open source software, in particular, a very popular open source software library called Deep LabCut, which is used for the analysis of experimental data from the behavior of animals in neuroscience. And that software also bridges between ideas in machine learning, deep learning, and the kind of analysis that we need to do in neuroscience. Finally, Franco Pestilli from UT Austin will talk about the way that communities can be formed within platforms for data analysis. So Franco and his team have created a platform for data analysis. And this sort of bridges us maybe into the next phase of data analysis or practice where people interact with very large scale systems for analysis of neuroscience data in cloud-based systems. And so with that, I'd like to thank, of course, all my sources of funding and people who've supported my work. You can check out the work that we do in my group at this website here. And without further ado, I pass this on to the speakers in this session.
Hello, my name is Eva Dyer, and I am an assistant professor in the biomedical engineering department at Georgia Tech and Emory. Today, I'll tell you about our recent work in representation learning from neural activity through self-supervision. The work is called Mind Your Own View, or MEOW. So before I jump into the details of using self-supervised learning, I'll first start by just kind of giving a big picture motivation behind a lot of the work that we do in my lab. So we are interested broadly in developing machine learning methods and optimization approaches for being able to understand and interpret high-dimensional data sets, and in particular in the field of neuroscience. So some big picture aims are to study and compare neural function and structure. And we do that in the same brain or in the same data set across behavior, across tasks, learning, or across different brains or different networks. For instance, across artificial neural networks and real biological networks, or across two different brains that are experiencing similar activities or underlying stimuli or dynamics. And in the end, we're really interested in being able to use these ideas of forming and comparing representations to then compare or understand what happens as the brain progresses across disease. And so to do that, we're developing new machine learning methods, as I was saying before, for doing representation learning and understanding how to align and adapt those representations so that we can actually then compare the representations in different networks. And as part of our work within representation learning, we're really interested in the role of self-supervision or different types of unsupervised objectives that essentially bake in implicit regularization to the network. And so without any labels, the neural network is able to essentially build good representations that are tuned to the sorts of structures in the data. And so the question is, how we can, or our overarching question here in this work is how we can actually use self-supervision as a way to form representations of many populations of neurons, and so doing representation learning from neural activity data sets at the single neuron level. So first we'll start off by digging into the question of what is self-supervision. And this has been a very popular approach that's been used, especially in computer vision, but is now starting to really take off in other domains, natural language processing, and work in graphs as well. And so the general principle behind self-supervision, which I've shown here in a slide from Jan LeCun, which is focused more on temporal self-supervision, but in general, the idea behind self-supervision is to essentially build a new view of a given sample and ask the network to figure out what's common or what's shared across these different views or augmentations that you basically introduce into the data. And so the example shown here of different ways of doing self-supervision over time would be to basically, take a collection of data or take a time series in this case and mask out different parts and ask the network to be able to predict what happens, throughout the rest of time. Some examples here: predicting the past from the present, or the future from, recent or future past, predicting the top from the bottom within an image, and, essentially just summarize, pretending there's some part of the input that you don't know and predicting that. And the idea is that, this is unsupervised in the sense that you have control over masking or removing information from the data. And the network has to learn to fill in that missing data. And in the meantime, it has to actually build a good representation that should encode, information and structure within the data over time as well. So given the recent successes of self-supervision in these other domains, we are really interested in whether or not we could utilize these tools as a way of doing representation learning from the activity of many neurons. And so the sort of setup in terms of data is, as I said, we have many neurons that we're recording from jointly over time. So this gives us, in the case where we bin and count the number of spikes within a given interval in time, we essentially will have an estimate of the firing rate of all these neurons over time. And, the first question in terms of building out this method for self-supervision for neural data, using the sort of ideas that we just discussed in the previous slide that are, sort of well established, this does share a lot of resemblance to ideas of predictive coding that are also popular in neuroscience. And there, the idea is essentially the brain wants to produce a representation of inputs or stimuli that will allow it to easily predict nearby moments in time or what should happen next, essentially. And so, drawing inspiration upon this, we can essentially, to start, maybe ask the network to be able to predict nearby time points and the neural activity that's formed from within them. The challenge, however, is that because the brain is actually shifting and there's quite a bit of, what could be called domain shift in terms of the actual raw population activity of the neurons over time, this actually builds, and as I'll show later, a relatively myopic sort of view when we only look over these nearby points in time, which are sort of shown here with this idea of, looking at a point X, trying to predict or trying to, form a representation of X prime, which is nearby in time. And when we do this over just local windows, which are really all that we know that we should be potentially able to predict very well, right, just due to sort of temporal consistency, when we do this, this often will form very myopic representations. And so, as I said, I'll show later those results, but what we did is we took a step forward or went further by asking whether or not we could, instead of just asking the neural network to predict these local points in time, actually ask it to predict further or non-local points in time as well. So going beyond this sort of local view. Of course, the challenge with that is now you need to know which other points you should be able to predict, right? Because if we link distant points that are actually not related to the current state X, then that could lead to negative results in terms of the representations learned. And the sort of intuition behind this idea, or what we can think about, is if the original local sort of self-supervised approach is sort of, instant by instant interpretation of the world, then we can think about linking across these non-local views as being kind of recalling memories or dreams or imagination, things that are similar to what you're experiencing at X, but aren't within, the very local present, essentially. So this idea is of, local and non-local sort of self-supervision or prediction is then instantiated in what we call mine your own view, or this idea of mining views or other samples from within the data set and using those essentially as a way of building predictive relationships within this neural network. And so our idea is to essentially mine samples in the representation space and then use them as views for one another. So what I'm showing here is, we know that X and X prime can be paired because they should be hopefully predictive of each other because they're nearby in time. And what we want to do is essentially find an XM by looking within the representation space and asking, if there's another point in time that has a similar representation, I now want to form some prediction between X and XM prime as well. Okay, so that's the main idea behind our self-supervised learning approach and how we think about it as being applied within neural activity data sets. And really the general perspectives are quite of mining within the data set could be broadly applied. And so we do show the application both in time series or neural activity, and we also show that we also apply it to images and show improvement over other STOTA or state-of-the-art methods for doing representation learning with self-supervision. And so we think that this idea of view mining is actually a very general approach that can be used in many different domains, and we're exploring that further. And so the approach is built on top of one of the state-of-the-art methods now for representation learning using these sorts of what are called dual deep predictive architectures. And so the idea behind BIAL, or bootstrap your own latent, is shown here. And we build on top of BIAL with our approach, and so I'll first describe it. So really BIAL starts by taking an image, or in our case, an activity profile of many neurons at a single point in time. And then from that image, you'll basically produce two different views by applying transformations T and T prime to generate X and X prime. So examples of images are shown here of two different views of the same image. Essentially, in the case of images, there's a number of augmentations or classes of transformations that are used to create these different views that essentially will break a lot of the nuisance parameters or other structures within the images that aren't indicative of the underlying class. And then from that, you have these different views, X and X prime. Essentially, what you'll do is you'll feed those through two different networks that are mirrored versions of one another, and I'll describe in a little more detail what that looks like. But essentially, we'll feed those images through to produce some latent representation Z and Z prime. And then our goal within the BIAL objective is just to minimize, in this case, a normalized L2 distance between the prediction through Q of Z and Z prime. So essentially what you're going to do is you're going to try to form a predictor that can easily map between these two different augmented views of the underlying sample S, so going between Z and Z prime, and then you do this over all these different stochastic augmentations of the underlying sample. And now the key thing is, or the reason why this won't collapse, is actually through the fact that we're asking this network to be able to do this across many images and many examples and the same fixed augmentation or transformation class. And essentially, we're not going to optimize the parameters of this network, but only the top one, and this network will essentially be a momentum averaged version of the original network. This online network. And so essentially by producing a small perturbation between the two different networks, this can also essentially produce a way to, solve this predictive task while also ensuring that the overall representation space doesn't collapse to a sort of trivial solution. And so, yes, so this idea, which is called BIAL, has recently achieved state-of-the-art performance in image classification and a number of tasks on ImageNet and other complex natural images. And so we can build on top of this overall framework through this approach that I described called Mind Your Own View, or MEOW. And we do this by essentially adding to this BIAL objective, as we saw before, this additional term that now, through a separate projector and predictor, is essentially going to try to minimize the loss between views, which, as I described before, are actually formed from different samples within the same data set. So in this case, we're going to look at the representation by passing the data through our first projector to the next one, computing V prime, and now we're going to minimize the prediction loss between those two different samples. And the key thing is that when we form the views that we use for prediction in this last network, we're going to use it through just a simple stochastic nearest neighbors approach, where basically we start with a candidate sample, YM, we're going to pass other candidate examples through the target network. We're going to find YM's nearest neighbors, and then we're going to randomly sample one of them and choose that as our view, and then actually plug that through this secondary portion of the network as I described. All right. And so the main sort of, I guess, hyperparameter here is essentially this lambda, and that tells us how much weight we're going to place on our non-local or within or across sample predictions versus these augmentations or sort of more local views of the data, which have been generated through, in the case of neural activity, through these local nearby temporal shifts. The first task that we wanted to apply to was a relatively simplistic reaching task from non-human primates. And so in this case, our goal is to essentially decode the reach direction from one of 8 different possible target directions from neurons within the primary motor cortex within non-human primates. And so what I'm showing here are results from applying MEOW, BIAL, and another competitive method, competitor method, which is used in self-supervised approaches called SimClear. And we're also looking at the results of representation learning on an autoencoder as well in this case. So as I said, we're recording from around 100 neurons in the brains of 2 different non-human primates. So we have results for Chewy and Mihi across 2 different days in both conditions. We have 8 different reach directions, as I described. And so in this case, our goal is to be able to decode which direction the animal is reaching to from one of these 8 different possibilities. So in this case, what we find is that consistently we obtain, the highest performance with MEOW or using this local and non-local prediction strategy and view mining approach. But what's really exciting and quite interesting is in many cases, these methods actually outperform a supervised baseline. So essentially taking a multi-layer perceptron or a neural network and optimizing it to solve this task in a supervised manner. And so what we're seeing here is, in some of the cases as I'm showing here, like MEOW will consistently outperform supervised decoders. In some cases it doesn't. But also BIAL in many of these cases is, almost reaching this supervised level as well. We're, as I said, very excited about this because of its ability to outperform supervised decoders. So what this is essentially providing us with is a strong form of an implicit regularization that we can use through these latent predictive models that help to sort of, build instability into neural networks, and that allows us to effectively decode things like, in this case, reach direction. So now just zooming in a little bit further into how exactly we're building these predictions and what are the augmentations that we're feeding into the network in this case. And so we've tried a number of different augmentations for neural activity or spiking neural activity, and all of this is provided through our website or through our GitHub, which I'll show some the website at the end. And so some different augmentations that have been applied include this sort of temporal shift, so locally asking the network to be able to predict across nearby points in time of some fixed amount. Then in addition to this temporal shift augmentation, now masking or dropping out neurons and asking the network to still be able to predict the neural state in the latent space, despite the fact that we've actually dropped out different neurons. Which is also a sort of interesting and perhaps brain-inspired augmentation, because it makes sense that, we should be able to decode information about downstream things like, the task or reach direction or state, even from a subset of neurons within this population. We can also add a little bit of activity or noise through a sort of sparse and additive noise model as well. And we can optimize, these different transformations. And what we find is that consistently, things like the temporal shift and dropout will build the most important regularization into the network and really take us most of the way there. And by adding in some additional noise into the neural activity patterns, we can further boost the accuracy, a few percentages as well. But I think the main takeaway point here is that, really to achieve this sort of strong self-supervised performance, we need to ask the network both to predict across different points in time and also from dropped out or missing sets of neurons overall. And on the bottom, I'm just showing sort of ablations where we look at bile and meow across these different types of augmentations and how we can, in the end, by adding in noise, pepper, and these dropouts, we can actually achieve this highest performance here. All right, after applying these ideas to these 4 different data sets from non-human primate, the next step was to go and see whether or not we could apply this into different behavioral tasks and across different, in this case, different organisms or different animals. And so here I'm showing results now from applying this method to decode different sleep states from both rat and mouse, in this case in 2 different brain areas, in primary visual cortex and also in hippocampus. And here we have 3 different classes to potentially decode from the representations that are learned through meow and bile and the other approaches, decoding REM, non-REM, and also wake from the brain. Again, we find that we get solid and consistent performance that outperforms or hits supervised levels in both of these cases. So, and this is all, I'm sorry I didn't actually say this, but so the protocol or the way that we're actually comparing in this case is we're training the whole network using this unsupervised objective and these different temporal or dropout augmentations. Then we freeze the network and we're basically training a linear layer on top of the frozen representations. And from that, we're then decoding these different tasks. So essentially the whole network is trained in an unsupervised manner, and then we're asking for the quality of these representations in decoded or downstream tasks on the frozen representations. And what we find here is that similar augmentation strategies can be applied despite these key differences between data sets. We're looking at rat, we're looking at mouse. So I think, we believe that these temporal and dropout-based augmentations can be a powerful strategy that can be used in many different contexts. In addition, we can then test these results on image data sets as well. Here I'm showing results from CFAR 10, CFAR 100, and MNIST. And what we're finding is that we can establish a gap over these state-of-the-art methods like BIOL and SimClear using Meow through this view mining approach. Here are some examples over on the right of samples, augmentations, and mined views in these different cases. And what we see is consistently we'll find other examples that are from similar classes, but even when we don't, we believe, like for instance here, right, different, well, actually, sorry, these are the same cat, but I actually don't have an example here. But yes, so we do find that there are cases where we will link different images across different classes, but they're still linked through some other conceptual or other semantic content that's within the image. And so the mining is still able to build interesting and predictive relationships across these images. So in summary, I introduced today a new approach for self-supervised learning through a cross-sample prediction called Mine Your Own View. We provide code and examples of how to use this both on images and neural data sets on the website here in our in Nerds Lab GitHub. And so really the main takeaways is, Meow provides a way to find views or samples from within the data set to learn from. This provides a general approach for both images and time series. And I describe the applicability and utility of Meow on being able to do representation learning on multi-unit neural activity and the applications to decoding reach direction from non-human primate motor cortex, as well as sleep decoding within the rodent brain. And with that, I'll acknowledge all the people that really made this work happen. And so the lead author on the work, Mehdi Azabu, and all the other folks in the lab that helped with this work, and then our collaborators Keith Hengen at WashU in St. Louis, and then our collaborators at DeepMind, Mohammed Azar, Michelle Valco, and then we also have folks at Hopkins that have been working with us on these and related projects as well. Our funding sources as well. And with that, I'll end the talk, and I look forward to a discussion later on. Thank you so much for your attention.
Hi, my name is Franco Pestilli from the University of Texas at Austin. I will be talking about interoperability and cloud computing platform. More specifically, I will be talking about brainlife.io. What is brainlife.io? It's a public, open source, and free cloud computing platform that supports reproducible neuroimaging research. Provides integrated data management, pre-processing, and visualization. It connects code wizards with data gurus to the top supercomputers. Promotes data sharing, code sharing, and advances scientific discovery. These are the three pillars and the three communities that contribute to the platform: code developers, data generators and archives, and supercomputing and cloud providers. There's three primary data modalities that are currently supported on the platform: anatomical MRI, functional MRI, and diffusion MRI. From these basic three modalities, multiple data derivatives can be generated, brain volume and surface analysis can be applied on the platform, brain network, both functional and structural, can be created on the platform, and then white matter mapping, both macroscopic and microscopic properties of the white matter can be studied on the platform. So how does it work? If you log in and enter the platform, data organized into projects. Each one of the project is managed by the data manager. They decide access to admins and members. Data can stay private as long as the members of the project decide so. Data can be processed individually, one subject at a time, or it can be processed via pipelines that apply automated processing across all the data and subjects within a project. Data can be also visualized. Here is an example visualization of the visualizer that we developed within the project, but also we use visualization routines and software developed by the community. The BrainLife community has been growing now since its first release about 3 years ago. We have users, over 1,500 user accounts created and about 100 users actively every month. Users are, as you can see in this map, are distributed in different parts of the globe. Interoperability. Data archives, data standards, and software libraries are 3 key aspects of software and neuroinformatics development out there. And these are the 3 assets that BrainLife brings together. BrainLife connects archives and software libraries via data standards, so to allow researchers to be able to process data on the cloud. Data archives, we connect with multiple archives. We are in partnership with OpenNeuro, and we can you can move data from OpenNeuro to BrainLife and vice versa from BrainLife to OpenNeuro soon. We also connected with the International Neuroimaging Data Sharing Initiative and many more. We indeed use DataLad as a decentralized data management system that allow us to connect with openly shared data sets across different projects. Here's an example. Within the platform, you can go to the dataset tab. Here, all the data sets that are shared through DataLad appear. Here is a dataset from OpenNeuro, and you can import automatically this dataset, creating a project, selecting the type of data, and creating a new project or importing into an existing project. And this will automatically grab the data and move it into now a project of your use. Once the data is in the project, you can see both the metadata and the neuroimaging data have been moved. You can then process the data, explore the data, visualize the data using different, the major software libraries for visualization. So data from archive becomes actionable on the cloud platform and allowing researchers to happen. Software libraries are also the workhorses of the platform. DiPy has been one of the early partner for the project. FreeSurfer, one of the top used libraries on BrainLife, FreeFSL2, MRTrix, will connect on Workbench, MRQC, fMRI Prep, DCM2NI, NiPipe, VistaSoft from Stanford University, NiBabel, AFNI, and ANS. All these libraries and more are bridged together into different apps. From libraries on BrainLife become services. Here, going back, you can go to the app tab, and here you see where the services are. A service is an app. An app is a ready available processing stream within the cloud ecosystem that you can run on the different resources that are made available by the developers for running analysis. You can read instructions that the developer leave on GitHub. All the apps live on GitHub, and you can also monitor who's using this app. You can see there's different people in green that just successfully completed the app, and some of them are waiting for the app to run. You can run the app from within this interface, or you can also run the app from within a project, as I showed you before, through the process or pipeline tabs. In practice, software libraries come alive as cloud services on the platform. The second axis is data standards. We are compatible with the brain imaging data structure. This is one of the upcoming and dominant standards for sharing neuroimaging data. And BrainLife has mechanism for inputs data. Data from scanner can be, and DICOM, for example, can be uploaded to a secure system, EasyBeads, that is upcoming in the near future. And from EasyBeads, the data through a GUI interface can be automatically transformed into a Beads dataset. Once the Beads dataset is created, the dataset can be pushed into a project on on the platform and then analyze. You can choose an existing project again, or you can choose a new project. At this point, you can do data anonymization if necessary. You can correct any mistakes that the automated guesses of easy beats might have made. And you can finalize the transformation from DICOM set and to beads, and from there push into brain life. Also, there's mechanisms for taking data out of brain life into bead structure. Brain lives allow to publish your project. That is a bundle that contains both code or services as well as data. This is one example, one project and one release. And you can see here that if you want to download, you can copy automatically copy that script, and that script can be run locally on your terminal to download the beads version of these data sets generated on BrainLife. There's some level of lack of coverage of some of the data derivatives on BrainLife because some of the beads, for example, beads for connectivity, are still being developed, and we're looking forward for the community to complete that task. Data standards indeed is used by the platform for data exchange, both in and out of the platform. Data can be inputted and outputted via the bead standard. So interoperability is at the core of the BrainLife technology. BrainLife uses community-driven software libraries, some open source and some not open source. And BrainLife connects with major data archives and repositories and acts like the glue between archives, researchers, and supercomputers. Data standards and software are key to our success and the success of the platform. And here I want to thank our sponsors. BrainLife was funded by INCF through the Brain Initiative, and we then received funding by NIH, Microsoft, and we're supported by Pittsburgh Supercomputing Center, the Indiana University Supercomputing Center, and TAC Supercomputing Center. And I'm open to questions. Meanwhile, I want to thank all the project members and the collaborators and other individuals from the community that have participated in the project by testing, using the platform, reporting errors and bugs, and simply just using it for their research. There's been several papers now that have been coming out with us involved and without using the platform. Thank you very much.
Hello, everyone. Thank you so much for all these talks. That was very, very interesting. And now we have some time for discussion. Just kind of summarizing what we saw. I think we saw here work at a variety of different skills and a variety of different on a variety of different topics. And I'd like to maybe try to pull together some of the threads that I saw common across those three talks, and maybe one thing that I'd like to hear from all of you a little bit about is I think one thing that's clear about all these different talks is that it involves working in collaboration between different fields and with different communities. And I'd like to hear maybe what kinds of lessons you've learned about collaboration, about putting together these kinds of these projects that involve experimental work on one hand, computational work. How do you, I think one challenge that can arise in doing this kind of work is explaining what you're doing to people from outside of your community, and explaining this to people who are using methods that you develop or tools that you develop and so on. So I'd like to hear a little bit about lessons that you've learned from that. Maybe we'll start according to the order of the talk. So maybe I'll start with Eva on that.
Yes. So as you pointed out, I think collaboration is really key for a lot of the projects that were discussed today. And, as your introduction sort of discussed, in data science in general, we need very collaborative and interdisciplinary sort of approaches. So for us, we work a lot with electrophysiologists, or, people that are studying the brain at different scales and at different resolutions. And across different domains of science, right? Like the sort of, I guess, expected standards, or how you validate, or how you sort of like show that your approach is valuable, right, for a community. Like that's completely different depending on which scientific community you're a part of. So I think that, as you pointed out, I think the communication and then expectations and sort of establishing those standards is a key part of, I guess, first building a sort of trust, or just like, everyone needs to be able to describe what their expectations are for a given collaboration. And I think the other thing that we've definitely come into has been challenging for us is the different time scales over which, say, computer science, and maybe Mackenzie can also speak to this as well, because in computer vision, right, and machine learning, things are moving at very fast pace. And so I think building both tools for which you can sort of put out into the community very quickly, and still, kind of building on benchmarks and well-established standards, and at the same time, using those to like really create longer term scientific impact, and building, tools towards neuroscientific or other scientific sort of outcomes, right? It's like kind of staging all of that, I think, is the challenge, but it's one that we're trying to work through now, and build some I don't know, kind of insights into how to push from both of these different angles. So those are
Yeah,
some.
maybe I'll move this on to Mackenzie. What were sort of lessons that you've learned? People take things that you do and maybe they expect them to do certain things. I imagine that people look at this and they say, well, maybe I can bend it in a little bit of this way and use it for something that it wasn't necessarily intended to do originally. How do you deal with that? And just in general, what have you learned for sort of these kinds of
things? First of all, thanks for this really exciting panel. It was a really excellent set of talks. It was very fun. So thanks again for inviting me, Ariel. Just to piggyback on what Eva was saying, I think, these communities can often have different goals and also have overlap in interesting ways, right? So as you mentioned, thinking about collaborations between even computer scientists and biologists, like the timescales that you're talking about, right? Like a lot of the computer vision works on conferences and benchmarks and shared data sets, and that hasn't necessarily always been the standard in, say, systems neuroscience. I mean, there are conferences that people push for, but not like the, everyone knows exactly when the CVPR deadline is, and that, that's like on the calendar. And it's not like having this deadline for pushing papers is actually just an interesting and quite new thing that I'm experiencing. So that's been a lot of fun as well. I think that's also added some cool energy into it. And I think there's also an interesting trade-off between let's say maintaining open source code and giving the users an experience, and who your user base is a really big point. And so I'm sure Franco can also talk to this. So for example, like what we're trying to do is really build on, state-of-the-art computer vision, right? We built on human pose estimation. There's established benchmarks so we can test networks. We did some transfer learning tricks. We made it be able to work quite well. But we have to, then maintain it and keep it up to state-of-the-art. So a lot of the research is actually kind of behind the scenes, like keeping this really state-of-the-art performance on these classical benchmarks, but at the same time, animal pose has its own new challenges. So I think as someone who works kind of with one leg between these spaces, or trying to, it's also been really rewarding for the computer vision community to also recognize that like animal pose estimation is a really interesting question and poses different challenges. And so there's actually more crosstalk now in these communities than there has been in the past. So that's, I think, getting really exciting. And just lastly, exactly what you were saying, it kind of gets back to this notion of like who your users are. And so, sometimes it's a bit, it always surprises me what people end up using this software for, which is really fun. So in the early days, we were very excited. Like it worked on animals and not just, I mean, humans are animals too, but like small furry animals that are, grayscale images. And there's a lot of other things about laboratory data that's quite different than natural image statistics. But yeah, people have used it for all kinds of things, like, tracking cancer cells, or in physics papers for tracking particles that I just wouldn't have even thought to do. And so that's been very fun. And I think in that sense, it's been very rewarding from the open source developer side to see, like, there's 1,000 works of our software. I don't even have the time to go see what everyone is changing it and bending it in new ways. But I think that's been a really fun part, is that, some of this gets put back into the main package, and it sort of shifted a bit of the research that we've done. Like, my lab is really kind of focused on motor control and adaptation, but at the same time, we've worked hard for the last few years on, building new tools for multi-animal pose estimation that might not actually even affect my direct research program that much, but it's been really fun to, like, think about new challenges. So that's been a great, that's been a great part of doing this. I'm not sure I fully answered your question, but hopefully I wasn't like totally tangential.
No, that was great. That was very cool to hear that. Franco, maybe to you as well. Sort of what do you have a very large number of people all over the world. Some of them you probably never get to meet and talk to. And how do you manage their expectations in this kind of context? How do you manage just like the onslaught of people showing up and doing things? And maybe you could talk a little bit about what you've learned from that.
Yeah, it's a complex question, and has multiple levels to it. I would say that the primary challenge, because of what we're trying to do with the project, which is to move labs to the cloud, it's actually a middleware problem, right? So if you go down to the folks with the libraries developing software, they understand what we're doing, they understand the terminology. Sometimes the word pipelines, workflow gets really confusing for us, many of us, because we use everywhere, but maybe that's one of the few. Then on the other side, the general public, I think it's easy to communicate to the general public, when you're talking about something what we're doing, because it's simple, you can abstract to a very high level. The middleware, the labs that have been doing neuroscience for a lot of time, running experiments inside labs, etc. That's a more complicated, that I have questions and interest to the platform coming from very different perspective. People come and say, hey, I would like to dump here some images that do microscopy and see what I can do with the visualization you guys have, which is a fair question, but it's a very challenging question on multiple levels because of the size of the data, the type of, the visualization goes with the data, etc. At the same time, challenges are in communicating why we're doing what we're doing and what is the value of what we're doing, and that's an ongoing conversation with other colleagues. It's like why we stop running on local computers, and and then it's a matter of discussing tracking of behavior, tracking of mistakes, helping reducing mistakes, and it's a constant, for us, it's a constant struggle. And I think it's part of what we do. I go back to your people infrastructure. I think actually our project is not just about technology. It's not about hardware, it's not about software. Part of what we do, we're very active Slack channel. Please join it, brainlife.io/Slack. And we're kind of creating some culture there so that people understand how to and why and what. So as you do, and also we do, we have workshops that we try to organize that is part of the cultural change, within the field, but also outside of the field, to understand why these platforms are coming and what we think can be doing for society, for the good of society. I hope I answered.
Yeah, that was great. Thank you. Yeah, there was one specific question from the audience. I'll convey it now to Mackenzie, a question about using deep lab cut and pose estimation more generally with clinical disorders. The question specifically was, have you worked with clinicians assessing or treating cerebral palsy or other motor tremor disorders, Parkinson's and so on?
Yeah, that's a great question. So of course, the field of human pose estimation, there's just so much great work in this space. And yeah, we've been lucky to apply some of our tools and others to these types of questions. My lab directly doesn't do it, but I've You know, that's another really beautiful thing about being an open source software developer, is that a lot of people, you know, kind of open up their world to you. And so we've seen a lot of really cool work. You know, I think we just hit 200,000 downloads of our software this month. And so it's just a massive, yeah, thanks, Eva. It's just a massive user base, but it's been really fun. Like there's actually some clinical trials ongoing that are using Deep Lab Cut that I've had no conversations with the people, which I think that's the coolest thing, right? Like people can just run with it and do this stuff. But we're actually, yeah, we're very much interested in particularly looking at these applications of, you know, just broadly pose estimation and video-based analysis to get better behavioral descriptors versus these sometimes very simple scales that are used to kind of make a better approach to diagnostic and treatment of things like cerebral palsy and Parkinson's and stroke. But yeah, my lab directly doesn't do it, but I'm super happy to see these tools in general being used in this direction. So I think it's fun. And maybe if I can like completely piggyback off this question to the rest of the panel too, I think this also raises this really interesting question. I'd be curious, especially from you, Franco, and in your experience about like moving things to the cloud and doing this with like video-based, you know, data is really exciting, but also raises a ton of interesting privacy concerns and like cybersecurity concerns. And so I'm just wondering how you're thinking about it in this space. Because I think with any of us like using AI, we have to sort of face these ethical questions all the time. And I'd be really curious to hear of your experience, because you certainly handled much more patient data than I have ever touched. So.
Shall I go?
Yeah, please. Yeah, if that's sorry.
So, well, we're taking the long route. We are actually not handling now data. We're focusing on basic science and on open data sets. We do as much as we can anonymization by following the standards and by doing dephasing, even though we probably all agree that anonymization or pseudonymization, it's a relative concept. You know, there's probably going to be a limit that is an algorithmic limit more than a hard limit to that. And yeah, so I had been in touch with collaborators that use more naturalistic type of stimulations and data collected on children inside homes, etc. And it's a challenge. There's technology for enclaving. There's technology for differential privacy. And I think all these technology are probably going to become part of the next generation of platforms we'll be developing. I think in a certain way, Brain Life is a pilot for us, even though we really believe in it. It was like, hey, let's try this and see how it works and how we can build this. And, you know, there's a whole dimension of how do you do the computing? How do you do the moving of the data? Like all the engineering that goes behind that. That was really our goal for the first, you know, the first platform like this that completely open. And then, you know, we're going to go back to do science maybe that is more encompassing of the different subdiscipline within neuroscience. So hopefully cover. Normally what I do in the lab, I reach out to collaborators that are experts in the field and try to work with them and like pose the needs that we're trying to face. One good thing of doing, you know, what you do and what we're doing, or kind of building software, is like you have this dimension of engineering in the project. So, and as you do that, you can build on the past, right? So, and then you can create a new direction by looking at your product, not just your results, not just your scientific result, but your actual software product has, you know, a route to the next challenge. So that's how we're doing.
Yeah, no, that's very cool. Thanks for indulging my my query.
I have a question to Eva about your method and about something that occurred to me while looking at that. Is that self-supervised learning in the context that you're using it, it's used as a way to sort of find a way to a good solution to a particular problem. But I'm wondering whether it can also be used in sort of online experimental design, right? Where instead of asking which sample should I be learning from that already exists in the data, rather you might ask something like, which is the next sample that I should get to best provide information in the context that I'm in right now.
Yeah, that's a really interesting connection between I would say these sorts of augmentation-driven approaches for self-supervised learning, and this idea of like actually wanting to find maybe a generative model that would allow you to resample from your data set and put forth kind of like candidate views or other samples that you might want to probe next, right, within an experimental design or some other sort of adaptive sampling sort of setup. I think that it's a very interesting idea to be able to use just representation learning systems in general to then, of course, you know, if you have a good representation, you could use it somehow to generate new candidate samples, for instance. But I think, you know, one of the kind of key limitations, I guess I'll just go ahead and reveal it, right, within all of these methods, is that they rely heavily on the choice of augmentations that you use to sort of build in these invariances or build in this sort of regularity into the solution, right? And I think in the case of images, we know a lot about different augmentations that we actually do want to apply to images to build an invariance, like translating, rotating. We have a lot of domain knowledge that's sort of built into those underlying systems. Similarly, I think in temporal data, which is what I showed as well, right? Like we do know, okay, there should be some regularity in time if you're looking at a video or if you're looking at a time series. But what I think this has really revealed to us is that in domains where you don't necessarily know, you know, like what are the right ways to perturb a neural time series. I mean, we tried some simple things like using dropout and using these temporal augmentations. But I think, yeah, just when we're trying to apply these ideas in general in new domains, the choice of augmentations or how we actually build in the prediction challenges that we feed into the network become very critical. They're going to, you know, impose certain bias or regularities into the network. And if they're not selected correctly, then you can show, even in the cases of images, sometimes these approaches, you know, will push you in the wrong direction. And so I think the promise, as you said, of using it in these other contexts is really exciting. And first kind of figuring out how we can do this in a sustainable and kind of informed way, especially in new domains, I think is going to also be a critical first step to get there.
Okay, thank you. I think we're reaching the end of our time here. Thank you so much for this excellent set of talks. I really enjoyed that, and for this great discussion. Thank you so much for everyone watching us, and INCF for organizing the conference, and hope to see you all soon. Take care.
Thanks, Ariel, for organizing.
Thanks, Eva. Thanks, Franco. Take care.
Thanks all.